It's great to see everyone here, and I'm thrilled to introduce our next speaker, Dr. Masataka Nishiga. Dr. Nishiga is an instructor at Stanford Cardiovascular Institute. As a physician-scientist, he focused on unraveling the mechanism of cardiovascular disease and developing innovative treatment. After completing his cardiology fellowship in Japan, he pursued a PhD at Kyoto University focused on the role of microRNAs and non-coding RNAs in heart failure. Currently at Stanford, under the guidance of Dr. Joseph Wu, Dr. Nishiga's postdoc research leverages iPS cells and the CRISPR technique. His primary research area include the cardiac impact of cancer therapies and the cardiovascular effect of COVID-19. He recently received a NIH K99 award. Please welcome Dr. Nishiga.
Thank you for the kind introduction. Today, I'd like to talk about our recent project studying the mechanism of myocarditis after messenger RNA vaccination and its potential solutions. So, as we all know, messenger RNA vaccines have been a game changer in our fight against the COVID-19 pandemic. The Nobel Prize in Physiology or Medicine last year was awarded to Dr. Calico and Dr. Wiseman for their pioneering work in the messenger RNA modification. And the scope of messenger RNA technology is not limited to COVID-19, but it is expected to grow in many other fields, including vaccines for other infectious diseases and anti-cancer immunotherapies, gene therapies, and so on. The messenger RNA vaccines designed for COVID-19 have been safe in general. However, there are still rare adverse events, such as myocarditis. This side effect was initially reported from Israel, and it became a public health concern because the cases were predominantly observed in children. These are two initial papers in New England Journal of Medicine reporting myocarditis cases after vaccination. As you can see in these figures, both papers are showing that the increase was mainly after the second dose, as you can see here and here. And in particular, the patients were predominantly in young males in their teens or 20s. So, and according to recent meta-analysis studies, the average age is about 16 years old, and 90% of patients are male. And in clinical testing, most patients had elevated cardiac troponin I in blood test. And on the other hand, ejection fraction was basically maintained in most of the cases. In terms of the incidence, the overall incidence was quite low, but there is a significant difference between the first dose and second dose. The majority of the cases are after the second dose. So the typical clinical scenario was young boys and a few days after the second dose. This myocarditis was quite rare, but if you consider the global scale, this is still a concerning adverse effect because we have been vaccinating more than 5 billion people in the world. Also, this myocarditis may be a class effect in any type of messenger RNA vaccines. So investigating the mechanism of this myocarditis is still important to enhance the safety of future messenger RNA vaccines in many fields. And our project was inspired by this paper from our collaborators at Stanford. They studied time course response to Pfizer vaccines in healthy volunteers. As you can see here, the peak of the immune response was 1 or 2 days after the second dose. And this time point was exactly the same with a typical onset of myocarditis cases. So this led us to hypothesize that this myocarditis phenotype is a result of hyperimmune response. And recently, a mechanistic study was reported from a group in Yale University. They used blood samples from myocarditis patients after vaccination. They showed there is no evidence to support the mechanism of autoantibody against cardiac proteins or allergic response to the messenger RNA vaccines or overproduction of anti-SARS-CoV-2 antibodies. So they denied these possibilities, and instead they showed the elevation of many cytokines in these patients, as you can see here. So basically their findings are also supportive for our hypothesis. And in our project, first we tried to identify cytokine mediators. We combined the two studies of human data I showed. Here, x-axis shows the cytokine levels of myocarditis patients compared to control from the Yale University study. And y-axis shows the cytokine levels of day 1 after vaccination from the Stanford study. And here we found that interferon gamma, among 27 cytokines that are detected in both studies, the interferon gamma and its downstream cytokine CXCL10 were consistently upregulated in both conditions. And second, we conducted in vitro experiment using human iPSC-derived macrophages. We exposed these macrophages to Pfizer vaccine or Moderna vaccine and measured the level of cytokines in the supernatant. And in the supernatant, interferon gamma was upregulated by Pfizer vaccine, and CXCL10 was upregulated by both Pfizer vaccine and Moderna vaccines. And the result was consistent with the human data. And one interesting observation here was that TNF-alpha, which is one of the most well-known cytokines, was not induced by vaccines. And this was also consistent with the human data. As you can see here, TNF-alpha is here. So these data suggesting that the response to messenger RNA vaccines do not upregulate generic inflammatory pathways, but specifically upregulate interferon gamma pathway. So these two cytokines were upregulated after the vaccination in human. Next, to test whether these cytokines can damage cardiomyocytes directly, we treated human iPSC-derived cardiomyocytes with a cytokine cocktail of CXCL10 and interferon gamma. We used two concentrations. The low concentration mimics the serum level after vaccination in healthy individuals. And high concentration reflects the higher level of these cytokines in myocarditis patients. So first, a marker of cardiac stress, NT-proBNP in the supernatant was increased in a dose-dependent manner when we treat cardiomyocyte with the cytokine cocktail. And in terms of the function of cardiomyocytes, the contractility was impaired by the cytokine cocktail, and there was a dose dependency as well. We also measured electrophysiology phenotypes using MEA, and the high concentration increased rhythmic events in MEA, and field potential duration was prolonged by both concentrations, and there was a dose dependency as well. So these data are suggesting that these two cytokines are damaging cardiomyocytes directly. We also checked the gene expression levels by RNA sequencing. The cytokine-treated cardiomyocytes showed a myocarditis-like gene expression pattern with many inflammatory genes upregulated. And here, we noticed that proteasome-related genes were significantly upregulated as a unique feature of this cytokine-induced cardiac injury. So this is indicating a potential mechanism of enhanced degradation of cardiac proteins in these cardiomyocytes. So the next question we had is how we can prevent this adverse effect. And as I mentioned earlier, there is a significant sex difference in this myocarditis. 90% of the patients are male. So we thought estrogens might be playing an important protective role in female. So in this context, we picked up a chemical compound called genistein. The reasons were this compound has an estrogen-like structure, and it is known to have anti-inflammatory and cardiovascular protective effect. This cardiovascular protective effect was previously shown by our previous study, in which we performed a computational drug screening and identified this compound as a protective compound against marijuana-induced endothelial inflammation. So based on these points, we thought this compound, genistein, could be a promising candidate to prevent this myocarditis adverse effect. So to test the effect of genistein, we treated iPSC-derived cardiomyocytes with genistein and then exposed the cardiomyocyte to the cytokines. At the gene expression level, the expression of cardiac stress marker, BNP, was rescued by the treatment of genistein. Also, interferon gamma responsive genes, such as STAT1, IDO1, were also rescued by genistein. And at the protein level, phosphorylation of STAT1, which is the main driver of interferon gamma downstream genes, was also rescued by genistein treatment. We also check the protein level by using mass spectrometry, and inflammatory proteins and proteasome proteins were increased by cytokine treatment in cardiomyocytes. And these proteins were downregulated by the treatment of genistein. And we also saw some of the cardiac structural proteins, such as myosin light chains, were downregulated by cytokines. And one of the cardiac, these proteins, which is myosin light chain 12, was rescued by genistein. So this result is supporting the potential mechanism in which cytokine treatment enhance the degradation of cardiac structural proteins, and genistein can mitigate the degradation. So next, we measured the activity of immunoproteasome, which is a type of proteasomes activated by interferon gamma. Here, ONX is a specific inhibitor of immunoproteasomes. So we are using ONX as a kind of positive control in this experiment. As you can see here, immunoproteasome was activated by cytokine treatment, and it was completely blocked by ONX. And interestingly, genistein treatment inhibited the activation of immunoproteasome like ONX. And more importantly, at the function level of cardiomyocytes, cytokine treatment impaired the contractility of cardiomyocytes, and ONX rescued the impaired contractility. And genistein also rescued the impaired contractility as well. So these data suggesting that the overactivated immunoproteasome can be a therapeutic target. Lastly, we performed an in vivo experiment. First, we conducted a pilot study using mice to see whether this transient surge of these two cytokines can cause cardiac damage in vivo. We injected these two cytokines from the tail vein and checked the level of serum cardiac troponin I in a time course experiment. The level of cardiac troponin I increased immediately after the cytokine injection, and the peak was around from 2 to 6 hours after the injection. So this result is indicating these cytokines can cause acute cardiac injury in vivo. And also this small pilot experiment is showing that the transient increase of these two cytokines, which was observed in humans after the second vaccination, is sufficient to cause cardiac damage in vivo. So based on this initial pilot study, we set up 3 groups, including the control group, cytokine group, and the cytokine plus genistein group. These mice received genistein or vehicle via oral gavage for 1 week before cytokine injection. And on the experiment day, these mice received 1 single administration of cytokine cocktail from the tail vein, and the blood and the tissue sample were collected at 4 hours based on the previous pilot study. So in histology, we detected infiltration of immune cells in 4 hours, such as macrophages into the heart in cytokine-treated group, and genistein rescued the infiltration of macrophages. And finally, the main readout, cardiac troponin I, was increased in the cytokine group, and which was rescued by the treatment of genistein. So these in vivo data are confirming that genistein has a mitigative effect in cytokine-induced cardiac injury in vivo. In summary, there is a transient surge of interferon gamma and its downstream cytokine CXCL10 here, which is immediately after the second dose of COVID-19 messenger RNA vaccination. And this time point exactly matches the typical onset of myocarditis cases. In our in vitro and in vivo experiments, these two cytokines caused cardiac injury. And based on the significant sex difference in clinical cases, we came up with a chemical compound called genistein. And this compound was able to mitigate the effect of cytokines in cardiomyocytes as well as in vivo mouse heart. And lastly, since the application of messenger RNA vaccines is expanding to many areas, we hope our findings can contribute to enhancing the safety profile of future messenger RNA vaccines. I'd like to thank the lab members of the ULab for their support of this project, especially Shu Kao, who conducted the main iPSC experiment. I'd like to thank all the collaborators and my funding sources. With that, I'm happy to take any questions. Thank you.